Welcome back to Science in Parallel, a podcast about people and projects in computational science. I'm your host, Sarah Webb, and in this episode, you'll hear from Anda Trifan and Amanda Randles, computational scientists from different fields who both work on big questions in biology and medicine using leadership class supercomputers. Their research projects have attracted some of the highest honors in high-performance computing. Between them, they've been finalists 5 times for the annual ACM Gordon Bell Prize or the recent Special Prize for COVID-19 research. Their common ground doesn't stop there. Both received the Department of Energy Computational Science Graduate Fellowship, and they worked on urgent pandemic research projects while raising very young children. We discuss the scientific questions that drive them, the adrenaline rush of round-the-clock COVID-19 research, and their words of advice, particularly for other women in science. Anda Trifan is a graduate student at the University of Illinois Urbana-Champaign who uses molecular dynamics simulations to study the interactions between proteins and membranes in cancer and other diseases. Through her Argonne National Laboratory practicum, she joined the teams that modeled the SARS-CoV-2 spike protein and virus aerosols. And Anda was first author on a third paper that simulated part of the SARS-CoV-2 replication process. All 3 projects were finalists for the Gordon Bell Special Prize for COVID-19 research, and the spike protein paper won the 2020 award. Amanda Randles is an assistant professor in biomedical engineering at Duke University. Much of her work has focused on using medical data to build a 3-dimensional image of an individual's unique geometry of veins and arteries. That allows them to simulate large-scale blood flow toward building tools that could help doctors diagnose and treat disease. Some of that research for their blood flow simulator known as Harvey was a finalist for the 2015 ACM Gordon Bell Prize. Amanda has received numerous other honors, including the ACM's Grace Murray Hopper Award in 2017. Welcome, Anda. Welcome, Amanda. It's great to have you here.
Hello.
Yeah, thanks for having us.
One thing, looking at, say, your CVs, for example, is that, Amanda, I know that, say, your undergraduate training was physics and computer science. Anda, yours is in chemistry. But in many ways, it seems like you've ended up working on very similar types of problems. So I want to get from each of you the development of your interests and how you landed in this computational biology space. You'll hear from Anda first.
During my undergraduate degree, I studied chemistry, specialty in biochemistry, and I really enjoyed that. I love doing experiments in wet lab. But after my bachelor's degree, I worked in industry for a while for a Dow chemical company, and I didn't feel like that's what I wanted to do for the rest of my life. When I looked at PhD programs, I looked at very interdisciplinary options because I wanted to test out what else is out there and what I would enjoy doing. So, computational work has always been really fascinating to me because you're able to immediately visualize the things that you're working on. So, especially molecular dynamics, you get to do science at a resolution where you see in much detail what's happening in a biophysical system. So, at University of Illinois Champaign-Urbana, of course, they have one of the best programs in molecular dynamics where they develop NAMD and VMD, and those are used across the country. I ended up working with Professor Ahmad Tashkhorshid, and in his lab, we work on proteins and specifically protein membrane interactions and things like that. So I really enjoy being able to do things at that level and see atoms interact with each other and how that affects a certain system.
I think it's your turn, Amanda.
Mine is somewhat pretty similar. When I started out, I was my main degree in undergrad was physics, so I was taking the really trying to understand the fundamental side of what are the equations and how do we capture it. And then I was also doing computer science, but similarly, I went to industry before going back to graduate school. I worked at IBM for a while, And when I was there, I got to work on the Blue Gene system, which is where I first, really got exposed to parallel computing. And I got really excited about, not just building the supercomputers, which is what we were doing more at the IBM side, but how do we actually use them and what kind of questions can we solve that you wouldn't otherwise be able to? So that was what prompted me to go back to graduate school. I ended up getting my Ph.D. in physics. So I've always been kind of on the physics computer science side, but then taking that to solve biomedical problems. So I really I've always been passionate about the medical questions and the biomedical application, but take a physics-based approach to how do we develop these models. That was kind of what brought me into the project I'm working on. I started in graduate school. We're doing physics-based modeling to capture fluid dynamics using the large-scale computing skills and kind of bring all of that together to work with the clinicians and answer clinical questions. And so it's been that merging of different fields, I think, is really interesting throughout.
Can you each talk to me a little bit about the biological questions that drive you and how computation fits into those questions?
I started my research lab in 2015. I had been at Livermore as a Lawrence Fellow, and while I was there, they were also incredibly supportive, and they let me put together a workshop right before I left where we just invited people from all over the country just to come talk about research, and they would just come. But we brought together researchers from cancer biology. We had interventional cardiologists fly out. So we had people who are experts in visualization. Anyway, we had people from all over and bring them together and really go over. We talked about Livermore. We had people from the big computing side and such of the capabilities. And we've really just had a 3-day brainstorming session of what are the key challenges in our areas? Where could we really, make an impact? And out of that, we ended up coming up with a few key questions in cancer research and in cardiovascular work that ended up really being the major projects I started my lab focused on. From a physicist standpoint, it's always hard if there are many questions I think are really interesting and you tell it to the cardiologist and they're like, that's great, but that would not change how I would practice and is not necessarily a useful question. So it's been really helpful to have the clinician there to say how do you actually practice? What are you doing? There are a lot of invasive diagnostic tests. Where could we start? The first step was one of the main projects that came out of it was current practice was using coronary angiography instead of CT to determine if someone needs a stent. And they had just the fundamental question of could we use coronary angiography to create 3D models? Because coronary angiography are 2 pictures of someone's heart. Can we use those images to create a 3D flow simulation so that instead of having to put someone in the cath lab, insert a wire into their heart to measure the pressure in their heart, could we use a simulation to accurately measure the pressure gradient across a narrowing in your vessel and determine should you or should you not place a stent? We just finished a 200-patient clinical trial where we're able to show that you can actually do, we just finished the clinical trial now, and this has been, 7 years since this question first came up. It helped kind of identify some of the key questions in that area and then bring to that, using these computational models, we can get much more complex measurements and get more complex phenomarkers, describing the hemodynamics. We're looking at quantities like vorticity, which is describing the turbulent nature of the flow. You can't measure that easily directly with a guide wire, but you can get those characteristics or shear stress on the wall or other complex metrics using these simulations. So now we're looking into questions like, the vorticity, are you more likely to be able to predict adverse events in 2 years? Can we find more complex markers that you can really only get out of the simulation to then guide treatment and inform diagnosis on that end? So I think those kinds of questions are really exciting of what can we pull out when we couple an experimental setup or an in vivo measurement with these simulations, and how do we add to and augment that data? On the other side, we also were interested in when you're running a fluid structure interaction model, you're looking at cells. Simulations give you that unique capability to change one factor and hold everything else completely the same. And you can run the same experiment. It's different than running something in vitro where the humidity in the air may change. It's very hard to exactly replicate exactly what you had done beforehand. But in a simulation, we can change just the bending stiffness or just the size of the nucleus of the cell and see how, pinpoint how changing this one quantity is going to result in, behavior of that cell and really sort out what is the contribution of that one factor. So I think that's really interesting and exciting if we can, it's a unique way of looking at data that simulations kind of bring that you can't get in other ways. And so we were trying to figure out, thinking of how do we really use that and how do we learn more about, what is it about certain cells that cause them to stick to different regions in the body, and how do we actually learn more about cancer cells? Why is it clusters? Why do clusters behave differently than single cells? And really trying to get into those questions were the things we were initially, trying to think of. And of course, that also brings into play all of the computational questions of, if you want to look at a large number of red blood cells, like you need a large scale. Like you can, it's very computationally intractable to model a large number of cells on a few nodes. You need leadership class systems, and that brings out all of the interesting high performance computing questions that kind of come into play. How do we use that data? We've also had a few, like we had just started at that time looking into how do you actually use virtual reality to couple in with your physics based simulations to try to have more intuitive interaction when you're doing treatment planning. How do you drive simulations with that? There were a lot of just those kinds of questions we were dealing with ahead of time.
So, Anda, talk about your work kind of early on.
Actually, as soon as I joined the PhD program, I said that the one thing I really don't wanna work on is cancer because, everybody works on cancer, and it's just absolutely so complex that I don't think studying very small systems could, elucidate anything new in any way. So but of course, that's what I ended up working on. I worked on a very small protein called RAS, and it has 3 different isoforms, and I specifically worked on KRAS. This is this is a signaling protein, very important in our bodies, and mutations in its active site are responsible for about 98% of pancreatic cancers and many others like lung and other cancers. So, one very important aspect of its function is that it functions at the surface of the plasma membrane. And so its interaction with the membrane is best characterized using molecular dynamics simulations because many times experiments kind of don't give you enough resolution to really see what's happening there. So when I started working on it, there were no treatments and no really hope in sight. But since then, Amgen actually came out with a drug to target one of the mutants, where one of the amino acids is mutated to a cysteine, and so they developed a drug to bind covalently to that and to help kind of keep this protein in its active state or deactivate. And so, my simulations were of the wild type protein, which means the regular, no changes to it. And then I also studied the mutants, which are the ones that cause problems and help this protein stay in an active state, which means it keeps signaling and then cells keep growing, and that's what causes the cancer.
I want to ask you both how COVID shaped your research. And Anta, I'll start with you because it sounds like that was very much a turning point for you in a very critical time in your PhD. And talk to me about how you got involved with COVID and what it was like to work on the biggest problem of our time.
I did my first practicum with Arvind Ramanathan at Argonne National Lab, I 2020, and I was supposed to finish in April, and March, the pandemic hit. And so, of course, as with everybody else, everything just kind of dropped and everybody started working on coronavirus related things. And this was a week before my practicum ended. So technically, I could have just left, but I was so curious about how the virus worked and why it's so easily spreadable and why it's so dangerous and how it affects all of our organs. And so none of that was known at the time. We know of SARS-CoV, the original one, and MERS, but this one has such high infectivity rate, and I was really curious about how this is different and why. And so I asked Arvin to join his coronavirus projects right away. And he was like, yes, of course, we can use all the hands that we can get. So I started working on smaller systems first. With COVID, there are two main proteases. There's MPRO and PLPRO. I worked on the smaller one. This one kind of cuts up the viral RNA and then it translates it into the subsequent proteins that need to be used in the viral replication cycle. But aside from that, we got involved with Romy Amaro's lab, who built the first virus system. And we also collaborated with Lillian Chong's lab from Pittsburgh, and they do this enhanced molecular dynamics method called WESPA, weighted ensemble simulations. Lillian's lab studied the spike protein and specifically the open to close transition. And that's very important because that's the first point of contact that the virus has with our ACE2 human receptors. And so this open to close transition was very important in trying to figure out, how exactly the virus binds first to our cells. And so they did that part, and then Romy's lab did the full simulations of the spike, and then also did the simulations of the spike binding to the ACE2 human receptor. And what we in Arvin's lab did was take all of that data and use machine learning to extract very relevant and important information from them. Because typically, when you have hundreds of nanoseconds or microseconds of simulation, it's very difficult to kind of sift through it visually or with any kind of analysis tools and extract what might be interesting or relevant at a biophysical level. So everybody kind of played a different part in this project, and this led to our Gordon Bell submission. And eventually, we won with that project in 2020. Yeah. And then last year, the pandemic was kind of tamed by having the vaccine, but people were still getting infected. So for me, it was very interesting to see what happens post infection. And so I started studying the replication transcription complex. This is responsible for replicating the viral RNA and making more viruses within our cells. And we did some work on trying to simulate how RNA is being transcribed within this complex. And also, it has a mechanism to backtrack, which means it finds errors within the RNA and corrects them so the virus can keep replicating. So we simulated the backtracking mechanism, and currently I'm studying remdesivir, which is one of the only drugs that have been approved so far for post-infection. And I want to know how it works at an atomic level because it did work for SARS-CoV, and that's why it was repurposed for SARS-CoV-2, but nobody really knows why it stops this replication mechanism and how it aids in symptom relief sooner than, placebo. So currently I'm doing simulations of remdesivir within the RTC, and it's such a complicated name. RNA dependent RNA polymerase and how it functions. So it's been quite the ride since 2020. And really, there's so many things to be studied about coronavirus and so many proteins that are crucial to its function and how they work together. So I've kind of had a taste of working on everything from the outside of the virus with the spike and then its replicating mechanism as well.
So did you have experience with the machine learning side of that before the coronavirus projects, or was that brand new? And
No, that was all brand new for me. People from NVIDIA, and they're the experts in actually writing the code that we used, but I was able to apply it and extract the biophysical, meaningful things that we were able to do. So in our project, everybody kind of had their own role. The people that did the coding and that side of things were not experts in biophysics, so they were able to write the machine learning part of it and hype the HPC. They were able to optimize that, but then they weren't really able to interpret the results, and that's where my expertise came in. And we looked at the different conformations of the spike, and we looked at how, for example, one of the things that we extracted from these simulations was the stalk of the spike bending, which was seen in experimental results, but we would have to have milliseconds or microseconds of these simulations going in order to observe that also computationally. But with machine learning, we were able to speed that up by like 10 or 11 times because we were able to pick out some of these rare states and then restart simulations from there instead of having one very long trajectory where we would need to bring it to that experimental observed point. So it was extremely interesting and such a honor for me to be able to work with all these incredibly intelligent people in our field. So that was probably the most exciting time in my career so far.
So I have one more follow up just 'cause I think this is really interesting. Like my experience with the pandemic was like it brought people together to work in a way that like you actually collaborated quickly and
Yes.
effectively. How did you do the handoff between the computer scientists and the biophysicists? How did you guys actually do that and bring together people from NVIDIA with, the lab academic researchers with it?
Yeah, so I was in an actually like a pretty unique position because I was closely collaborating with both the CS people and the biophysics people. So for me, it was really exciting because all of these people were located geographically in very different parts of the world. And so Zoom enabled us to really work together because anytime we had any questions, you would just jump on a call and it was very effective in communicating our issues. So I don't remember much pre pandemic how it was, but I feel like it wasn't as easy to communicate with people across the country. Yes, you would make appointments but I feel like the pandemic really, really just enabled scientists to work together faster and more effectively. We learned how to communicate much more effectively and much quicker. But I did feel that immediately after we started talking about the Gordon Bell submission and how things just really progressed very fast. So I think we decided to do the submission in July or August, and then much of the work was done before individually. So like the steps were taken in Romy's lab, they had the simulations, and Lillian's lab, and then we had kind of a backbone for what we wanted to do with the machine learning part. But really, this collaboration started very quickly and rapidly before we had to submit in November.
Yeah, it's a phenomenal having so many different groups coming together to work on one project from like, you couldn't have started earlier than March and then you had a finished product by November. It's a very unique setup.
I really, really enjoyed it. I have to say it was like adrenaline rush all the time because something was due at any point.
Right. Right.
As anybody working in science knows, it's not a given that you get any meaningful results from any kind of experiment. So it was all just really exciting to be able to contribute to science and at the same time to kind of merge all of these different methodologies together and come up with a project that was so relevant at the time.
Especially as a graduate student, it's a very exciting opportunity, but it's also I feel like that's very brave. You don't know how that's going to impact your graduation or how, you have the path to the PhD and this is a very like it could pay off or it could just, delay
Exactly.
things.
So I honestly, I didn't even think about any of that at the time. Like to me, this project was so interesting and the problem that we were trying to solve was just so fascinating that I didn't even consider whether or not would be part of my thesis or if I'm even allowed for it to be a part of my thesis, but I was completely willing to work on it, separate from everything that I had to do in grad school because I was learning new things, and whether or not they were directly related to what's gonna end up in my thesis, it didn't really matter because these are skills that I can apply at any time during my career, and the collaboration with so many really smart people for me was probably the best aspect of it because I learned so much from so many different fields. So even if this meant delaying my PhD by 6 months, I was willing to take that. But for me specifically, it was even more intense because at the time I had a 5-month-old baby. So a lot of the meetings that I was in, I was either like with her on my lap or, she was like screaming in the background. So it added to kind of like the chaos and the adrenaline of getting everything to work. Also, it helped that I had to feed her, like, throughout the night because I was up working at hours that
Yeah.
other people weren't.
One of my questions was what working on this problem at this moment where obviously life was jumbled too was like for both of you. And Anda, I think you've spoken to that, but Amanda, I'd like to sort of get your perspective on that. And I also want to hear about your COVID work too, but I think we're in the life jumbling aspect of COVID. What was that like?
No, like mine was very similar. We had a COVID project we jumped into, but I have twins that are 2 years old now as well. So like I had like 6-month-old twins. We worked on trying to like how to split ventilators and trying to address the like, lack of ventilator issue that was really hitting in like March and April in 2020. And like the first meeting we had with Microsoft to work on that, I had like both twins sitting with me. And like it was very much like one would start crying, I'd pick her up, and like, they'd see the one crying one, she'd stop crying, I'd put her down, then the boy would start crying, and it just like rotated the entire meeting. And it was just like, yeah, nice to meet you all. I like this. This is how it is. I did not run all of the simulations. It was definitely the students that were running more, but I was like up on Slack, feeding at 2 a.m. Like, okay, I can see how the jobs are doing and like check in with you. Like it was weirdly help, like not like it was like a little helpful at times of like you were up at odd hours to be able to check on things, and it's like I'm here around the clock because that's just what it is right now. Yeah, it was definitely more interesting. And,
I do think that's another unintended consequence of the pandemic. This realization that people have personal lives, and our personal lives have become much more intertwined with our professional lives, because when you're at home, you're not necessarily able to separate those very well, especially, with your kids at home. So I feel like as a graduate student, that's not really known well among my peers, because I'm literally the only one in our lab who has kids. But I feel like as you advance in your career, PIs and postdocs, researchers have kids because they're at that point in their lives. And so it became a lot more acceptable to have your kid in a meeting, or, have to stop for 5 minutes to whatever, give them a snack. I don't know.
right.
So
Yeah, we had it. It was really interesting. Even like the Microsoft team we worked with, one of the leads there actually had twins. So like this never would have come up in
right,
any like
exactly.
normal working environment. He ended up like giving me advice.
Do you also feel like this social aspect of our lives have been a lot more pronounced since the pandemic?
Yeah, I think it's, I don't know. You people talk about it more. Like we had just little things of like one of the articles about us on the ventilator project. Like there's a picture of my whole family in the article, which is not normal. Yeah, everyone with young kids kind of, it gets, you either don't have a daycare slot, your daycare is closed half the time. Like it didn't, it kind of continued. And I think it's really gotten people to be much more understanding and kind of talking about like, well, how are you doing? Everybody's a well-rounded person, and it's okay to have these other sides.
Yeah. I also think it helps women in science a lot, because we don't want to seem as if our kids are taking time of our lives and we're all just really focused on our careers, but that's not really true, right? I mean, you do have to split your time, and you do have to cater to all of these different aspects of our lives. And so I think it just brings to light that we do so much on a daily basis that was kind of, ignored or pushed to the bottom before.
Yeah.
So we're seeing that other people are also kind of struggling with their work-life balance, and it's okay to do that, and it's part of life. And I'm really glad that it kind of was brought to surface, because it's not easy sometimes.
I think there's so many ways in which this time was hard, and it reshuffled how we think about everything, and it's just really interesting to hear the 2 of you kind of talk about that. But Amanda, I wanna be sure we talk about your ventilator work. You were here managing twins, and you have an older child too, right?
Yeah. So we have a 4-year-old now, but yeah.
But you got involved with COVID too. Tell me a little bit about how you got involved and what you worked on.
It was right in the beginning when, we were hearing everything about like the ventilator shortages in the US. And we had previously worked with some people on the medical side at the Duke campus, and they really started a project trying to figure out how do we make the ventilators we have go further. So they were trying to find out ways to split ventilators between patients that didn't necessarily have to be matched. So they could have, different weights, different properties. They were questioning if you could use flow simulation to figure out how do you actually tune the ventilator and create the product to split the ventilator. So they kind of came to us and asked if we take our blood flow simulation, do an airflow simulation of the ventilator to try to help them tune their model. It all happened really quickly. Like that team pulled together in March. Like before we even got together, they created a brand new tool that could split the ventilator, be 3D printed, and be accessible to hospitals around the country. And then I asked my students, if anyone wanted to be involved, who wanted to help. And we ended up creating 3 different teams that were trying out different models and figure out what we needed to do. From the scientific end of everything, all of our experience modeling blood flow is not necessarily that helpful in converting to airflow. Students created a brand new model. They had to validate that model. They worked with the team to do like benchtop comparisons with mechanical lungs. Like, it was pretty fantastic of just like, they created a brand new model within 2 or 3 weeks and validated that using tools we had never used before. We got to the point where we had the simulation, we had it validated, and we wanted to have all of the data available so we could put it in for FDA emergency use review and have it available for the doctors. The idea was the doctor would put in information about the ventilator, that they happened to have the 2 patients they were trying to match, and then it would tell you how to tune the apparatus that this team had created. The weight of the patient, height of the patient, ventilator, it kind of explodes quickly with a number of parameters. And so we joined in with that HPC, the consortium for COVID that came together early March, April of 2020. We ended up working with Microsoft, but working with people from, completely different backgrounds. It just came together with no ego, no questions about IP. People just jumped in immediately and wanted to help. The part I think is really interesting about this story of we submitted the application to the consortium on a Monday. We were paired with Microsoft by Tuesday, Wednesday. We had our first call with Microsoft on Thursday. And then we ran all of the simulations we needed by that following Monday. And we ran 800,000 hours of compute time in the cloud that weekend.
That's incredible.
No one slept that weekend. And it was researchers from Microsoft who had no idea what they were signing on to, stayed up all weekend to help, monitor this and make sure it worked. The first meeting we had with Microsoft, they brought in fluid dynamics experts in case we needed that help. They got in touch with people from MATLAB to try to figure out some issues we were having to get things to scale on the back end. They really pulled in anyone and everyone to make this happen quickly. We kind of approached them and we're we know we could be smarter. You don't have to do a brute force. Let's model everything. But our dream would be, let's just model everything and get it done. And they just gave us the compute hours and helped us finish it. That part was really amazing. We didn't end up getting emergency use approval because, the pandemic moved on. We didn't end up needing it. I think it's good information to have. We have the models out there and it's helpful to have for the future. And it's a good thing it wasn't used. Yeah, it's an interesting side of it.
That's just super cool.
Yeah, it's very, very exciting.
Ours was much shorter. It was only a month and a half, but it was very much the same. Very high adrenaline. How do we everyone was kind of sitting there, you want to do something and you want to help. I think it was good to try to feel you're productive and helping in some way, because I think everybody probably felt a little helpless at that time.
Yeah.
And
I wanted to get your perspective on what that's like to kind of be both at the cutting edge of the science and the cutting edge of the computing. For example, do you ever get frustrated by the computers not quite being able to model what you want them to model yet?
I love trying to figure out how far can we push it with the next biggest system. We're part of the Aurora Early Science Program, and it's super exciting to just sit and brainstorm of well, what can we do? But then there have definitely been a few moments. There was one where you forgot an exponent, and it was oh, we thought we could go to a 1 micron resolution. It was nope. The next biggest system, we can go from a 9 micron resolution to an 8 micron resolution. It's that is much less exciting. There are questions it is physically intractable to do a brute force simulation of all the red blood cells in the body. That is not going to happen on today's computers tomorrow. We need different algorithms, we need different methods. It's not just let's throw more processors at it. And that, really making sure you're accurately bounding your problems and understanding yes, we have access to the biggest systems, but that doesn't mean you can do anything. And lately I feel for a long time we played with questions in situ visualization, and it was more of it's fun to visualize while you have it, but not necessarily necessary. And now we're at the point where the data is a real issue. Yeah. Even, Summit or the systems today, we're using the entire memory and then we're running 1 million time steps. So you're creating, let's say, a petabyte of data, and then you're doing that for 1 million time steps. You have to be processing on the fly. You can't make a video of the entire thing at the resolution you want to. We had a big push for a project last year where we were trying to get a picture of the shell of the data of what we were simulating. And that was on Summit. The person making the picture was at Livermore, and it was 450 gigabytes just for the shell. And it was just trying to figure out how do we get that to Livermore for the picture. We now have the capability to run these simulations, but actually, how do you analyze it and do something productive with it? It's not frustrating of what we can't do with the systems now, but it's more of we have the capability to do this amazing research, but it's hard to really capitalize on it and make use of it. And we're working a lot more of the advanced algorithms to make it, kind of shift the way we're modeling, and what kind of how do you tie in things like machine learning? How do you accelerate? How do you do those sides? I think a lot of what we've done in the last 10, 15 years has really been pushing, let's go to the bigger scale. And so we did the first model that you can take the entire supercomputer and run the full 3D human body. And that's great. like we can model one heartbeat. If you wanted to model, a whole week or an hour of someone's time, fundamentally, like you're never going to get all of Summit for 2 months to actually model even an hour of someone's life. So that we can hit the scale, how do we extend the time periods we can model, and how do we make the most of the time when you do get something like all of Summit? How do we make the most of that compute resource, and how do we make sure we get everything we need to out of it? I want to play in the space of you need the biggest supercomputer, and I think that's really exciting and fun. But yeah, trying to make that tractable and do things with it is where we're at.
So, Anda, for the kinds of things that you're working on, and obviously COVID was pushing Summit and what was available to the limit, I'm sure.
A big problem is, so now we're able to build systems like the aerosol, for example, and it's in the billions of atoms range. But one major issue is the amount of data that comes out of it, for sure. And so struggling with having to even transfer to be able to visualize any of those simulations, because we don't quite work on the petaflop scale, but still you have terabytes of data that need to be analyzed. And so one issue is, how do you manage transferring even any of that data from like a supercomputer like Summit to a local workstation so you can literally just visualize 5 nanoseconds of a trajectory. So that becomes a problem really quick with expanding systems. But another one is, okay, now we have simulated the virus in an aerosol. Where do you go from there? So what are the things that you want to answer, because most biophysical things happen on order of milliseconds or, microseconds. And realistically right now, we would need, Summit for months to be able to really simulate something of that time scale. So, of course, one of the first steps in any emerging technology is to be able to do it. So, proof of concept is always very important, and just being able to put these systems together and simulate them for 1 nanosecond is an incredible victory for our field. But from here until you are actually able to extract information that you can apply, let's say tomorrow, to a biophysical problem, I think we still have quite a ways to go. But again, we need tools like machine learning or just smarter ways to analyze these larger trajectories, because you want to be able to, like Amanda said, capitalize on being just the sheer fact that you're able to do these things.
What advice would you pass along about getting started in this field?
One of the things that I'm passionate about is getting more women in science, because in most of my meetings, I am one of the only girls or women in the meeting. I think one of the deterrents of seeing more women in this field is that a lot of us don't think that we can handle our personal lives and our professional lives at the same time in such a demanding field. And I would say from experience, and Amanda is an amazing example of this, that you absolutely can. And there's resources like other ones of us that have been there to rely on. And so I think it's very important to not give up your personal life for your career necessarily, or wake up 15 years later and regret that you didn't do what you wanted also on a personal plan. So specifically for women in science, it's very possible to do everything that you wanted to. Of course, it's insane at times, but I think it's worth it, and you don't need to sacrifice either part of your life.
It is very hard at times, but it's also, it's incredibly rewarding, and it is possible. And we're all, found it easier, like throughout the pandemic, of you can reach out. It has opened things up where, you're not just reaching out to people who live down the street from you. Like, it's been helpful to reach out to other female faculty members who have kids who have this and can give advice and are there, and like having this support network is like, I think we're all much more open to that, and really, it's okay to ask for advice of how to do it, and also just listen and know if you need to say that like it's hard and I just need someone to understand that it's hard. Like, I think it's been helpful to know that other people are finding it hard too, but like it is possible. And I think the big thing, I know that kind of resonates with me, that's kind of come out in both of our explanations, is like finding something that you're truly passionate about. Like, the COVID situation is very unique, but if you are truly passionate about it and excited about making an impact, like I think that is it is worth finding out a way. You don't want to look back in 20 years and be like, there were other things I could have been much more excited about. And you try to figure out how to work on the type of problems that really, get you out of bed in the morning and make it worthwhile to make this trade-off. It is difficult to try to balance family and work, but it's worthwhile when you have, amazing family time, and then also are, genuinely excited about the thing you're doing during the day. And
to build on that, one piece of advice that I would have is find a team where you feel supported and where you feel like your ideas or your work is being put forward and not pushed down. Throughout my career, when I worked with people who were supportive, I definitely was a lot more productive. And I mean, obviously that makes sense, but it's not always the case. So for me, that's a very important step when choosing a lab or when choosing a workspace.
Well, on that note, I want to thank you both for your time. This was such an amazing conversation, and I learned a lot, and I hope you enjoyed it too.
yeah,
So
thanks for including me.
To learn more about Anda and Amanda, please check out our show notes at scienceinparallel.org, which includes links to an article from the 2022 issue of Dexis magazine about Anda and a Microsoft article about Amanda's COVID-19 ventilator work. This episode concludes Season 2 and our 6-episode series exploring how pandemic events have shaped computational science work and workplaces. Please subscribe and rate us on your favorite platform, and we'd love to hear from you via email or on Twitter. Science in Parallel is produced by the Krell Institute and highlights computational science with a particular focus on work by fellows and alumni of the DOE Computational Science Graduate Fellowship. Krell manages this program for the U.S. Department of Energy. Our music is by Steve O'Reilly. This episode was produced and edited by me, Sarah Webb.